Welcome viewers. Today I am going to discuss about certain assays in fluorescence microscopy which have revolutionized cell biology. Now I will talk about fluorescence resonance energy transfer or FRET, fluorescence recovery after photobleaching which is FRAP, fluorescence loss in photobleaching which is FLIP, and fluorescence lifetime imaging microscopy which is FLIM or FLIM. Now, I will also talk about the use of electrons in illuminating objects and talk about the details of Transmission Electron Microscope or TEM and Scanning Electron Microscope or SEM. Coming to the fluorescence microscopy and the assays, let's also talk about fluorescence. Now, what is fluorescence? Fluorescence is an emission phenomenon. So, when an incident light comes and interacts with a fluorophore, which is a chemical compound, first the electron goes to its excited state or excited electronic state as I call it. Now once it goes to the excited electronic state, it now tends to come back to the ground state. Now this is known as a relaxation. A part of it is non-radiative, which is called vibrational relaxation, due to which it comes back to the least level of the vibrational level in the excited state. Now once it comes to the lowest vibrational level of the excited state, it now will come to the ground state. During this, it liberates a photon of energy, and this is known as fluorescence. Typically, the time span of fluorescence is 10 to the power minus 12 to 10 to the power minus 9 seconds. Unfortunately, there are certain phenomena which may happen due to which there can be decrease in the intensity of fluorescence. Now, two such processes are known as quenching and photobleaching. Now what quenching does, it's a reversible process. Due to interaction of various chemicals, for example acrylamide or iodide which is around the fluorophore, it causes a decrease in the quantum yield of the fluorophore.
What do you mean by quantum yield?
Quantum yield of a fluorophore is actually the fluorescing capability of a fluorophore. Now this Q is equal to the number of photons emitted by the number of photons absorbed. Now coming to the another way by means of which there is loss of quantum yield other than the quenching is photobleaching. Now photobleaching, unlike quenching, is an irreversible process due to which the area where the excited electrons are happening get totally damaged, and that is the fluorophore get totally damaged and the fluorescing capability is lost. Now coming to the assays which are used in fluorescence microscopy, I will first discuss about fluorescence recovery after photobleaching or FRAP. Now in FRAP, what happens? First, a particular region of interest in the tissue or in the cell is selected, and intense laser is used to actually bleach that region. So let's say this is a whole region. Now laser is used in that whole cell to bleach it. Now once that region is bleached, then the camera is still on and the microscope still works, and we see how is it getting recovered. Now what happens in cell, if there is fluorophores which are hovering around with attached to proteins or which are attached to the plasma membranes, then if they diffuse in, it will cause the recovery. Now this actually has opened up new vistas of understanding the dynamic nature of the plasma membrane. Now the second type, which is also a part of FRAP experiment but the readout is different, is known as the FLIP, which is the fluorescence loss in photobleaching. If you photobleach a region, then the fluorophore from other part comes to that region due to diffusion. So that other region, there will be loss in the fluorescence intensity. In case of FLIP studies, we actually take that loss. So fluorescence recovery after photobleaching and FLIP are complementary to each other, but both can be used to study these phenomena in cell biology. The next assay which I am going to discuss is fluorescence resonance energy transfer or FRET, which is also known as Foster resonance energy transfer, depending on the person who actually invented it. Now, in case of FRET, what happens if there are two fluorophores which are so close together that there can be dipole-dipole coupling, then one fluorophore acts as an acceptor and one fluorophore a donor. So the incident light is accepted by the donor fluorophore, which get excited. Now, this will lead to corresponding excitation of the acceptor fluorophore, and this will happen when this excitation energy is liberated by the donor. Now, this principle was first introduced in 1940, where the rate of transfer Kt is equal to 1 by the fluorescence lifetime tau d into 6 power of the ratio of capital R0 by R. So what are R and R0? R0 is known as the Foster radius, which is the distance between the donor and acceptor when the efficiency of energy transfer and its probability is 0.5. And R is the distance between the donor and the acceptor. Now, what are the major conditions of FRET? Can both fluorophore or any two fluorophores act as fretting pairs? Now, this is very important to know. In reality, not all fluorophores can act as fretting pair. The first important condition of a fluorophore to be a fretting pair is that the two fluorophores should have some overlap of their spectra. What I mean by that is the emission spectrum of the donor fluorophore should have an overlap with the excitation spectrum of the acceptor fluorophore, due to which the energy is transferred into the acceptor fluorophore. The second most important thing is that the fluorophores should be in a very small distance to each other, and this distance can vary from 1 to 10 nanometer. And I should also tell you that because of this, it helps in providing extra resolution, and this is a very important assay to see protein-protein interactions. The third important point for fretting to occur is that the fluorophores should be properly oriented. The efficiency of energy transfer in case of FRET is equal to R0 to the power 6 by R0 to the power 6 plus R to the power 6. What we can see that this FRET technology can be used to identify interaction of different proteins which are tagged to fluorophores.
Sir, I have a question. What are the compounds that can be used as a fluorophore in FRET?
Now, very good question. So let's take an example. If I take a calcium calmodulin or even some other proteins, I will tag one protein with a CFP, which is cyan fluorescent protein, and the fretting pair for CFP is YFP, so I will tag the other interacting protein with YFP. Now, as you look at the figure, a particular orientation of these two proteins should be there, as well as these two fluorophores should be there for FRET to occur. Now, you also look at the figure in the spectrum, as you see that the absorption spectrum of the first fluorophore, let's say CFP, so you have to provide the incident energy as per that absorption spectrum. Now, the CFP absorbs it and then gets excited, and while it fluoresces, rather than actually fluorescing, it passes its energy to the second fluorophore, which is the YFP, and you get fluorescence from the YFP. So what you do is actually you excite CFP and get fluorescence of YFP. Only when all the FRET conditions are maintained, then you can see the fluorescence from YFP. Now, FRET is widely used in biology to see in vivo protein-protein interaction, to see the dynamics, to see of the membrane, to see the lipid rafts, as well as to see multi-protein interaction and the trafficking of different proteins from one part of the cell to another. Another assay that is used in fluorescence microscopy is fluorescence lifetime imaging measurements, or fluorescence lifetime imaging microscopy, which is FLIM. Fluorescence lifetime is defined as the time the excited electron spends in its excited state. Now, that is the lifetime of a fluorophore. Now, this is the backbone of fluorescence microscopy. Another important aspect of this film study is that we are trying to study this lifetime of the fluorophore, during which the intensity of fluorescence is not of major concern.
Sir, what are the advantages of FLIM?
The major advantage of film, as I was talking, is that we are here studying the fluorescence lifetime of a particular fluorophore. So let's take an example. Let's take there are two different fluorophores at two different conditions in the cell. And the fluorescence lifetime is actually dependent on what are the boundary conditions, what are the things in the vicinity, how the ions are present and not. Now, if one fluorophore in a particular part of the cell where a pH is very high, the lifetime will have a particular value. The same fluorophore which is present on the other section of the cell, let's say a fluorophore has gone into some vesicle or has traveled into the Golgi apparatus or even some secretory vesicles, then the pH value is not the same. Here the lifetime will be affected, and this can be better detected in film studies rather than in studies of fluorescence microscope, where intensity of fluorescence is only calculated. The most important aspect of the FLIM study is that it can be utilized to check the transport of different particles in the cells and even the movement of vesicles or the dynamicity of the membrane. And how? Just by noting the lifetime of the fluorophore. Coming to an important aspect of microscopy is resolution. Now, light microscopy has its inherent problem that because of light use, the inherent resolution cannot go below 200 nanometer. So how do we resolve below 200 nanometer? Now, in recent time, some modification of fluorescence microscopes has been done to attain better resolution. However, for high magnification and resolution of around few angstrom can only be obtained by the use of electrons as a source of illumination. Now, this is the basis of electron microscopy. A typical electron microscope will have an electron gun, due to which the electrons will come and impinge on the object, and then it will get scattered or get reflected from the object, and that can be focused by objective lenses and visualized on a fluorescent screen. The first prototype of electron microscope was made in 1931 by Ernst Ruska and Max Knoll. It was Ruska who received Nobel Prize in 1986 in physics for his discovery of the electron microscope. Let's come to the different parts of electron microscope. Now I am talking of the major parts. The first and the most important thing is the electron gun. The electron gun has a V-shaped tungsten filament which is present in the cone of the cathode. Now, opposite to the cathode there's an anode, and you provide an electric current. Now, as an accelerating voltage is provided to that electron gun, electrons coming out from the tungsten pass through a small hole at the end of the cathode and moves towards the anode. This is the basic principle of how electrons come out of that gun. Now, more the accelerating voltage, the lower will be the wavelength of the electron, which vary from 0.025 angstrom and still below. So you can well understand that the resolution will also be in the similar scale, and people can get a resolution of up to 2.5 angstrom using a 200 kilo electron volts acceleration. Now, the other components of electron microscope are the lenses. Unlike light microscope, which has optical lenses, electron microscope has electromagnetic lenses, which helps in focusing. The first pair is the condenser lens, which focus electrons coming in stream from the gun into the specimen. Once it passes through the specimen, depending on its deviation and scattering, these are collected by the objective lenses and then an intermediate range forming an intermediate real image. This is then focused by another set of projector lenses into either the camera or a fluorescent screen. Another aspect of electron microscope is that high vacuum is maintained within that system. The sole reason of using vacuum is that the electrons will interact with air particles and might get deviated. To prevent this, we keep vacuum. Now, coming to the two major types of electron microscope, one is a scanning electron microscope and another is a transmission electron microscope or TEM.
Sir, I have a question. So what are SEM and TEM used for?
Now, SEM and TEM are not used for the same purpose. Now, scanning electron microscope are mostly used to scan the surface topology of different biological or other samples. For example, let's say the surface topology of a virus. On the other hand, transmission electron microscope, as the name tells, transmits the electron beam through the sample. So I can get details about the cross section, details about the internal structures of a biological specimen. Now, let's take a look at the scanning electron microscope or the SEM. Welcome to the Scanning Electron Microscopic Facility of CSIR, Indian Institute of Chemical Biology at Jadavpur, Kolkata. What you are looking at is a scanning electron microscope or SEM, and this is the Vega 2 LSU of T-scan. The most important part of the microscope is the electron gun or the source of the electrons. So, electron comes out from this with a definite accelerating voltage. It falls on the object or the specimen which is present on beads, and these are coated with heavy metal. So, this is, you open it and place the samples here, and after it, once the electron gets scattered, these are detected by the detector placed here, and it feeds to the software in the computer so that one can see the image. Furthermore, another important component of such electron microscope is the vacuum, and there are high speed turbo vacuum pumps which are associated with this machine, which maintain the high vacuum in the system. The problem with most of the scanning electron microscope is we have to maintain high pressure and high vacuum. However, in recent days, there is environmental SEM or eSEM where some hydration is also allowed and no coating with heavy metals is required, but high vacuum is required around the specimen. Now we are at the TEM facility. What you are looking at is the transmission electron microscope, which is the FEI Tech9 G2 BioTwin. The most important component here also is the electron gun placed at the top, and this is 120 kV. The electrons passes through the condenser lenses, and then you can put your sample in between, and then there is the objective lenses, and it gets focused on the screen which you can observe manually. In nowadays, they are also coupled to the computer by the use of a camera, and this can be controlled by softwares present in the computer. I should mention that the lenses are not optical but electromagnetic in nature, and vacuum is maintained using different pumps so that the electron beam does not break. One of the major problems of using TEM is that in case of biological specimen, contrast which is obtained due to scattering of the electron is very low. That's why the specimen has to be processed before taking to the TEM. Another important aspect of using TEM is higher magnification along with high resolution. The magnification which people can obtain can go up to 1 lakh times. As I was discussing, sample preparation for biological specimen is of utmost importance. So first, the sample need to be fixed. Glutaraldehyde and paraformaldehyde or a mix of this can be used. Even osmium tetroxide is also used as a good fixing agent. Now then, the sample need to be dehydrated by grades of alcohol and then embedded in epoxy resin to make the thick hard sample. Why it is done is that this hard sample can be taken to a microtome to cut very thin or ultra sections. Once the sections are cut, they are taken on a copper grid and again stained with heavy metals and then taken to the TEM facility for imaging. In case of some biological specimens, other type of sample preparations can also be done. One of the examples is negative or positive staining. In case of positive staining, lead acetate or uniline acetate can be used to stain the sample per se. However, in negative staining, we actually stain the background and not the sample. So, contrast is enhanced for some objects. Another type of sample preparation is freeze etching or freeze fracture technology. So, this is mostly utilized to study the various internal organelles of a cell. For example, I want to study the vacuole, I want to study the Golgi apparatus in an intact cell. I will freeze the cell, then cut sections with a fluorescent knife and then take it for microscopic analysis. In most recently, what people do is cryofixation for cryo-electron microscope. Now, one of the important advantage of cryo-electron microscopy is that the artifacts which comes due to sample preparation in conventional electron microscopy can be removed in most cases in cryo-electron microscope, where the samples are plunged in liquid nitrogen. They're taken, kept in very cold condition around, let's say, minus 100 degrees Celsius and taken to the microscope and observed directly. A good microscope like 200 kV microscope and cryo facility can actually help you see the interaction of proteins or even the structure of proteins with a resolution of around 3 to 2.5 angstrom. Now, coming to the concluding part of this session, what we have done, we have studied the different assays of fluorescence microscope. We have also studied the different electron microscopes which are widely used, for example, SEM and TEM. I should also mention that in this microscopic lecture series, we have discussed about the widely used microscopic techniques, and we have actually refrained from using very specialized microscopic techniques which are used in some laboratory or some research purposes.